Hello and welcome to the Nucleate Boston 2022 Activator Final Pitch Showcase. Yeah. We really appreciate everyone coming out tonight. We had a great turnout. Just want to acknowledge all of the teams that we had this year across both the bio and the eco tracks for just making fantastic progress and really showcasing all the progress they made earlier today with all of the pitches that happened across the various rooms throughout campus. Now we're going to focus on the final 6 teams, and you will get to hear them pitch and compete and win some prizes. And before we get started with that, though, we'd like to tell you just a little bit about Nucleate as an organization and some acknowledgments that we would like to hand out to our community that's been so supportive of our mission and our programming. So with that, I would actually like to now turn it over to my fellow co-presidents, Sufyan Abdelhuda, who will take you through the overview of our organization leading up to the final pitch showcase. So please welcome Sufyan onto the stage.
Hey everyone, thank you all for coming here today. It's so nice to be doing this in person again after 3 years and since our first year doing it in person. So it's incredible to see how far we've come as an organization, and whenever you see the big letters, you know you've done something right. So my name is Sufyan. I am the co-president and chairman of the board of Nucleate and also one of the original co-founders. Today I'll be telling you a little bit about Nucleate and who we are as a next generation educational organization. And maybe I should mention my main job is actually as a PhD student over at Harvard Medical School in George Church's lab focused on building next generation cell therapies. So we are Nucleate and we are on a mission to empower tomorrow's biotech leaders by educating today's academic trainees. So we are a 501c3 Massachusetts incorporated nonprofit organization as of a couple months ago. Actually thought the day would never come we'd get that letter from the IRS, but here we are. We have an educational mission. Everything that we do is free and open to all, so that's to say that we do not charge any fees for our programs, nor do we take any equity in the teams that participate in our organization. We believe deeply in our open access mission. And lastly, we are, at our core, trainee-focused. And at least as of this year, 100% volunteer-led, which is quite incredible considering that, as I'll get into in a couple slides, we now have over 500 individuals leading this organization around the world. So we execute on our mission by having a set of goals that really focus on culture and community. The first of which is to provide free and open programming so that we can democratize biotech education to learners everywhere. We identify, educate, and develop talent to really elevate the next generation of biotech professionals. In doing so, we spark new intellectual communities and we build bridges both within and across the biotech ecosystem. And as an organization, we focus on fostering innovation, really increasing the quality and the quantity of life sciences ventures spinning out of academic labs. And in doing so, keeping a key focus on increasing access by building a unified community, really focusing on outreach and making diversity a top priority for us so that we can make biotech education accessible to all and not just a privileged few in Boston or San Francisco or the biotech hubs of the world. So really over the past year, we've undergone quite an incredible evolution and really to serve the needs, the unmet needs, I should say, of the community around the country and around the world. And this really started with our not so humble beginnings within the Harvard Biotech Club as a program called Activate. The Harvard Biotech Club, Michelle, who's here today serving as one of their co-presidents, has been around for about 25 years. Really started with the mission back when biotech was considered the dark side. Still is among some faculty, by the way. Really with a mission of bridging the gap between academia and industry and academia. And we've taken that core mission and really evolved it as a global organization. Running a variety of workshops, courses, lectures, networking series, diversity initiatives, professional development opportunities, as well as continuing to grow our core flagship activator program and the reason for which everybody is here today. At this point in time today, we are a unified community of scientist innovators currently operating in 10 cities across the United States, continuing and now running the fourth year, celebrating the fourth year of our founding chapter and flagship chapter today in Boston, but also running operations in Baltimore, in the Bay Area, Houston, Los Angeles, New Haven, New York, Philadelphia, the RTP region, as well as San Diego. And we're currently undergoing an evaluation and selection process to move into phase 2 of our expansion so that we can move into some of the other emerging biotech hubs around the country and hopefully around the world. So follow us on LinkedIn in order to get those news in terms of our expansion efforts. And lastly, before I hand it off, increasing diversity is one of our primary goals and matters deeply to us as an organization. So Nucleate is dedicated to justice, diversity, equity, and inclusion. Just a few numbers to really highlight where we are at this point in time, and hopefully within the coming months, we'll be able to release a more in-depth demographics survey about our organization, not only leadership teams, cohorts, experts, mentors, advisors. We have big goals for the biotech industry. But even today, at this point in time, our leadership team is composed of 50% women and non-binary individuals. We are 25% underrepresented minorities, and collectively as a student bodies, our nationalities represent over 44 countries around the world, and that's with less than half of the organization actually filling out the survey, so the real number is quite higher. And so without any further ado, it's my pleasure to introduce you to this year's co-managing directors in Boston, Sam, Rebecca, Oliver, and Jeff, who have done a phenomenal job and will tell you today about this year's Activator program in Boston.
Thank you, Sufyan, so much. And it's so good to be here today with all of you for our final pitch event for the Nucleate chapter here in Boston. My name is Jeff Knox. I'm one of the managing directors for this chapter here in Boston of Nucleate on the bio side, where we focus on human health. And I'm going to tell you a little bit about the Activator program, whose teams you're going to see complete their journey today. So the Activator program is the program we've been running for about 4 years here in Boston. It's the one that Nucleate sort of originated with the Harvard Biotech Club, as Sufyan described to you. It's a program that helps form new ventures. So back in November, when we first put all this together, there were groups of business and scientific trainees who we sort of scoured greater Boston to find and helped come together to form new teams. That's the first step in our process. The second step is giving them a whole bunch of different types of mentorship covering clinical, industry, legal sort of support that helps them get on their way. That's our second step. And then we put them through a rigorous curriculum of workshops that cover all the different types of risk we think a new company might run into. Technical risk, market risk, IP risk, you name it. And we do that in concert with a whole network of experts who we're so lucky to have supporting us here in Boston. And that makes sure that the teams who leave our curriculum are not only with a working knowledge, they're armed with a working network of people that they can reach out to when they run into problems they haven't even realized they have yet. And we wrap up our program by bringing those folks all in front of you today. And we're so excited to watch them show you all that they've learned. We're really proud of the alumni who've gone on from this program. In about 3 years, we've created about 36 alumni who have collectively raised more than $150 million in external financing. They've generated more than 50 jobs across greater Massachusetts and beyond. And we've already had one exit as well, which I think we're very especially proud of. You can see some of the names on our slide here. Again, just very, very proud of the alumni who've gone on from our program. And we're very confident that this cohort is going to be no exception. It's the most competitive one that we've had yet, including the largest number of teams we've ever supported. Back in November, when we first put this cohort together, we reviewed more than 200 applications and collectively built a cohort of almost 30 teams. And those teams represent institutions across all of Greater Boston. So we're very proud of the folks who we're going to be seeing today. And again, can't wait to get them in front of you. But all of this we couldn't do without the support of a small army of people who have helped us put this on. The leadership team alone, graduate students volunteering their time, is almost 80 people here in Boston, and we have a whole network of other mentors and experts who have supported us who push that number even higher. And so I know a number of those folks are in the audience today, and we just can't say thank you enough for helping us put all this on. And yes, please give them a round of applause. And now I'll pass it off to some of my co-managing directors to talk a little bit more about the folks who have participated in our program this year.
Thanks, Jeff. So I wanted to take a moment just to really acknowledge our amazing teams this year. You'll hear from 6 teams tonight in the final pitch, but they really just represent a fraction of our 29 teams in total, which Jeff alluded to in his previous slide, who really went through our activator program fully and worked tirelessly to refine their pitches, which you heard earlier today. This is actually the first year, I'm really proud to say, that we fully launched the ECO program. We had 10 teams participate, and these teams represent institutions from all over the country, from Boston to San Diego to Los Angeles to the Bay Area. The BioTrack this year had 19 teams and really is expanding year over year, and these all represent institutions from the greater Boston area.
And so a key part of running an event at this scale for our nearly 30 teams was to have 6 different breakout rooms this afternoon. And this wouldn't be possible, as is a common theme, without a number of people. Very importantly, our 8 semifinal judges who, in the ECO track, brought a wealth of experience across investing, operating, and clean tech more broadly. Similarly, in our bio track, we had 8 different judges who came from around Boston, bringing experience in healthcare, life sciences, to provide feedback and judge our teams through the semifinal round. So we're incredibly grateful for your time, support, and participation in making this event such a success.
Awesome, and for our final pitch tonight, I just wanted to take a moment to thank our round 2 judges who are sitting in the front. If you don't mind just standing for a round of applause. So just to quickly introduce them, we have Peter Barrett from Atlas Venture Partners. He's been such an asset and great advisor to us over the years for the Nucleate program as we developed to where we are today. He's also a professor and senior fellow at Harvard Business School, where he teaches a course called Lab to Market. We also have sitting next to him Mira Trucia, who is a senior partner at Westlake Village Biopartners. She has extensive experience investing in and sitting on the boards of life science companies, as she previously had roles at 5AM Ventures and Roche Venture.
And last but not least, we have Dr. Ann DeWitt, who is a partner at The Engine and has just been a huge help to launching our ECO program, and now in our 2nd year judging our ECO track. And not only does she bring a wealth of experience in the clean tech and investment space, but also in the healthcare space as well, previously serving as a partner at Sanofi Ventures. And with that, I'm going to hand it back to Oliver, who's going to talk a little bit about run of show and logistics for this evening.
Thank you very much, Sam, Rebecca, and Jeff. All right, and now the moment you've all been waiting for, the Boston Final Pitch Showcase, the cutting edge of the Boston biotech scene. So I'm going to start just by overviewing the rules, and I'll talk a little bit about some of the prizes that are available tonight, and then we'll get right into the pitches, and it'll be pretty fast and furious, but I know you guys are ready. So the way this is going to work is each team is going to be given 10 minutes to do the pitch. We will cut you off. There's a timer right here, so I will be watching it. The judges, you guys will have 5 minutes for Q&A. I will sort of semi-moderate it and let you know when we're getting close to that 5-minute cutoff. The audience does not have any sort of participation in the Q&A at this time, so we would ask that you remain silent so that the judges can be heard and the questions can be answered. If you're interested in following up with the teams, there will be sort of an optional networking thing happening afterwards in the atrium, and we encourage you to walk up to the teams and learn more about their technologies. There's several prizes that are going to be available today. Some of them are going to be available to both bio and eco teams, and others will be given out just to a single team. So just to run them down really quick, we've got first place prizes for both the bio and eco tracks totaling $2,000 each. We've got a program award and the audience choice award for $500 each. So the audience does have a chance to participate in that. At the end of the pitches, we will flash a QR code. we encourage you all to scan it and fill out a short form that will then be summed and tallied to figure out the winner of the Audience Choice Award in each track. Additionally, we have some sponsored awards that are being given out tonight. The Hatch at Nest Bio Award, sponsored by Nest Bio Labs, is going to award 6 months of free lab space to a single team, so it'll be really exciting to see who's in the running for that. Additionally, we're giving out a Scientific Excellence Award and a Diversity, Equity, and Inclusion Award sponsored by Alnylam and Genentech, respectively. So with that, I will try and get out of the way, and we'll move on to the final pitches. So as you guys heard at 5 p.m. during the reception, we've got 6 teams lined up: Real Seafoods, Atacama, Parasite Bio, Tenacity, Ingel Therapeutics, 3DOE. And with that, I would like to ask everyone to give a huge round of applause for the first team, Real Seafoods. Let's welcome them to the stage.
Hi, everyone. We're Real Seafoods, and we are on a mission to create delicious cell-cultured fish fillets.
Start timer.
It's thick, it's flaky, and it's indistinguishable from its wild-caught counterparts. We need to feed a growing population, and seafood is a large part of that equation. Today, more seafood is consumed than beef, and this is expected to double by 2050 in demand. But at the same time, the demand is increasing, supply is decreasing. We've already outstripped most of our oceans, but of the fish that we do have remaining, 90% are being fished at or above maximum sustainable levels. So we clearly need alternatives to this, and aquaculture is one such alternative. Unfortunately, aquaculture can actually exacerbate the problem. It takes about 5 pounds of wild fish to create 1 pound of farm fish. Plant-based solutions have been long touted as a sustainable alternative, but it simply lacks the taste and texture to capture the consumer. As a result, it hasn't been widely adopted, and last year only represented $12 million of sales in a multi-billion-dollar market. We propose a new alternative, making fish from fish cells. Cell-cultured seafood is not only better for you in the sense that we don't have to extract from our oceans, it also can be made delicious and local, which means that we can preserve biodiversity and also reduce emissions. Not only is it better for the planet, it's also better for you. As a consumer, we can make clean fish that is free of mercury, microplastics, and PCBs. And because it's made in a sterile environment, it also has a longer shelf life. Now, this idea of changing our food systems isn't new, and it's a big idea. And brands like Impossible and Beyond have proven the market for alternative protein. But these approaches are limited. I mean, how many times have you heard someone say, I don't want to eat plant-based, I just want real meat? And consumers agree. In a study by Mintel, consumers who won't eat plant-based food, 51% of them said that they don't want to eat it because they just want real meat. Our vision is to create cell-cultured fish with 100% cells. And others have tried to get there. They are basically using plant-based scaffolds to create these hybrid products that are about 60% plant. But these approaches are limiting in its ability to deliver the taste and texture that you expect of fish. And not only that, they also don't speak to consumers who don't want to try plant-based today. We have the technology to create a 100% cell product, and we'll be able to reach a broader set of consumers by delivering the product they really want. And now's the time to jump in. So over the past 5 years, what we've seen is that first movers in the market have had to address each of these verticals. Reason being that the industry was nascent. No one else was working on these problems, and so developmental costs were high. What's changed today is that many B2B companies exist that specialize in each of these verticals. And what this means is that we plan to leverage this emerging infrastructure and partner with companies to get to product cheaper and faster. We can focus on our key value add, which is creating thick and structured fillets in the most efficient and scalable way possible. And that's what we believe is the critical bottleneck for the industry for the next 5 years. Today, cultivated meat is made using plant-based scaffolds and with cells in between. And these approaches are limited in terms of delivering that springiness and that mouthfeel that you associate with fish. Also, uniquely to seafood, consumers look to seafood for health, and they don't want a product that is ultra-processed with 40 unfamiliar ingredients. We believe that the best tasting fish will come from fish cells, and we have the technology to make a 100% cell product. The only way to do this is to leverage cutting-edge tissue engineering techniques. We're going to use alignment to drive texture in our product and vascularization to keep cells alive until they mature. These techniques will let us create a better product than hybrid alternatives. Not only can we make a better product, we are also scalable and cost-efficient. Our streamlined manufacturing process uses injection molding. And so with that, we can directly go from the bioreactor to a fillet. What this means is less capital expenditures as we scale up, and also a lower cost to produce. Using that sheet stacking method that we had talked about before, you can imagine a room as big as this auditorium filled with sheets. That's a lot of sheets to manage, and a lot of media that needs to flow over it. Because we're making 3D tissue directly, we have lower media requirements, which is the largest input of costs. Now, it's important to note, our technology is not specific to any type of species, but we are starting in seafood for this reason. Seafood presents a stable, high price point relative to chicken and beef, and it's also cheaper to make. Combined together, this means a higher margins and profit potential for us. Fish is easier to make because it is made up of 98% muscle cells. And if you think about that compared to the complexity of a marbled steak, what this means is that we're able to get to a structured product faster. The other thing that's unique to fish cells is that they grow for faster and for longer, and at room temperature. And this translates to less energy and lower media costs on our end. With this, we want to go after the $7.4 billion fish fillet market to start. And once we figure that out, even though that is significant runway already, there's no reason why our technology can't extend to chicken, beef, and pork in the future, and why we can't partner with other companies to help them create thick, structured meat. This is a problem that requires innovation across all fronts. We need a better product, we need scalable production, and we need to speak to consumers. And our team brings together the best of biology, mechanical engineering, and business. John and Rob, my co-founders, come from one of the leading 3D biomanufacturing labs in the world. John's PhD focuses on vascularization and alignment, and Rob's PhD focuses on mechanical characterization and the development of novel production processes. I'm Bev, and I previously helped a plant-based company launch in the US, and I will bring a scale-up and commercialization angle to the business. Our team is complemented by Jennifer Lewis, who heads up our scientific advisory board and has co-founded 3 startups based out of research from her lab. We are committed to building out this business post the Nucleate program. We're in the process of licensing 4 relevant patents from Harvard, have raised preliminary funding to get going on our prototype, and all plan to start full-time later this year when we graduate. Over the next 12 months, we'll be focused on porting over these proven cardiac tissue engineering methods to show that they can work in fish. And with a $500K investment, we'll be able to demonstrate that our methods work in fish through the prototype of cell-cultured sushi. We're building the future of seafood, and we're excited to have you join us. Thank you.
Incredible. All right, we'll go right into the judge Q&A, and you guys finished a little early, so we got some extra minutes on the clock. I'll hand it over to our esteemed judging panel. Can we get the judge mic live?
Hi, great presentation. I'm excited about this vision of the world that you're painting. I wonder if you could give, like, at a high level, like, if you're going to capture some portion of the fish fillet market that you had outlined, just the gross, like, magnitude, like, what does that look like in terms of at some scale? Pick 10% or whatever it is. Like, what are we thinking about? Like, what's the size and scale of, like, bioreactors, the printers, just as you're thinking about like at scale, just for the fish fillet, like what kind of system are we talking about? That would be great. Just take us through what you're going to need at that scale.
Oh, what the manufacturing system would look like?
Yeah, just kind of what do you think, like high level,
Yeah.
like in terms of bioreactor capability, 3D printing,
Yeah.
like
Yeah.
we're running printers
Yeah.
the size of the state of Arkansas, or we're running it like the
Exactly.
size,
Yeah.
like high level?
I
Thank
think
you.
a big part that I want to emphasize is that we're not going to be using printing. So John and I come from a 3D bioprinting lab. We're aware of the limitations with that process. It's not a very scalable process. It takes a lot of time per individual product. And that's why much of our focus is to take the techniques that we learned in 3D bioprinting, alignment of cells, vascularizing thick tissues, but move it beyond printing to something that is scalable, a technique like injection molding, which is pretty much arguably the gold standard of scalability for manufacturing. And the only way that we can go about injection molding these tissues instead of building it up in layers is through this novel use of vascularization. Otherwise, if we were to inject a ton of cells into a mold to create our tissue, it would quickly die. You'd have the middle of it, it would be mush. So the only way that we're able to rapidly jump to this 3D size is with that vascularization. And so we're definitely going to have those challenges on the bioreactor side that any other company in this space will have. Luckily, there's been a lot of innovation on that side. We're hoping not to do that alone. But our focus is very much on turning this biomass into a structured product. Thank you.
Thanks for the presentation. It is not clear how far along this path you are. Do you have any sort of pilot running now? You talked about vascularization and injection molding. Has that been done ever? And maybe also, are there others trying to do something similar to you?
Yeah, I could jump in there. So we recently raised like a very, very small amount of money through this thing called the Olson Venture Fund. And we're using that money to port over these methods that have been done in many different cell types, whether it's cardiac, kidney, neuronal. We're trying to port these over into a fish muscle context. And we're actually have like secured a lab space and are trying to work on those proof of concept experiments now. And in the beginning, the kind of transition, we're not just gonna like make an injection molding process, right now. We're gonna be able to separate the process from these experiments of alignment in these cells by using printers in the absolute kind of like intermediate stage to prove out these methods and then scale those results into injection molding process.
Hi, great presentation, and I think you touched on one of the key questions I always have, which is, how is this able to scale and bring down costs? And you touched on some of the interesting features here that could bring down the cost of media, et cetera. Can you quantify that to a certain extent? I mean, compared to sort of what a bluefin tuna costs, assuming you were able to generate the same general mass of that meat, is this a twofold difference, a 50% difference, a tenfold difference as we start to think about really where the COGS, say, come in?
Yeah, I think you can try to, everybody's trying to reduce costs in many different ways. And already, it's come down orders of magnitude, and so now people are starting to get even more clever, right? We've moved beyond just, okay, this was optimized for CHO cells from biomanufacturing, and now it's optimized for these muscle cells. How can we get more clever than that? One example, for example, is there's ways around kind of bioreactor limitations. So right now, bioreactors, when people grow cells on microcarriers, the maximum packing fraction, how much cell you can pack into each bioreactor is, so if you look at different analyses online, it's like 0.35. So we've talked to different kind of startups in this space where if you could shield those cells into kind of like these encapsulated hydrogels, you shrink wrap them, you could increase that packing fraction up to the maximum of 0.64. So just like that's one example where you could like double your yield with the same amount of media. And there's different strategies as well, but that's just one example.
Great, thank you.
So what do you need from a team perspective, both today and like in 3 years? Like talk about the team build. What are you missing? What do you have? What do you want? Yeah, what's it going to take?
Yeah, so I think the expertise that we would need to add pretty quickly is a geneticist that can help look at genetic engineering techniques that will help us make this scalable and at a price point that is accessible to the average consumer in the long term. And I think the other thing to consider too is, as much as this is a technical problem, this is also a business problem. And so we need to start thinking about building out a lot of those partnerships that we saw earlier when we say we want to partner with B2B companies to test out trials to see, is this realistic? Can we scale with you? Or do we have to build this ourselves? And I think after we answer those questions, we can define for ourselves what type of capacity we need to build in-house versus we can outsource.
Great, we've got time for maybe one more quick question, if there is one. Okay, awesome. Well, please give it up for Real Seafoods, everyone. Thank you very much. Congratulations.
Thank you. Stage right.
All right. Oops. Okay, guys, we're going to move right on here to the next pitch, which is Atacama, our second Eco Track finalist. And they're working on some really exciting naturally compostable plastic films. So without further ado, please welcome Team Atacama.
Thank you. Hello, I'm Jose.
I am Paloma.
And thanks to Nucleate for this opportunity. We're excited to present to you our company, Atacama Biomaterials, named after the Atacama Desert in Chile, in that picture. We develop materials for a sustainable world. Unfortunately, the Atacama Desert looks like this right now, flooded with plastic waste. This horrific reality inspires us to act, because the problem repeats all over the world and it's even affecting our health. During their lifetime, everyone in this room will ingest an equivalent to 70,000 plastic bags in the form of microplastics. Governments, companies, NGOs, researchers, entrepreneurs, a lot of people in this room are looking for solutions, but it hasn't happened yet. Most importantly, people care. I care, and I'm pretty sure you care. Packaging has become a major concern for consumers. Yet, current alternatives, as the one I have in my hand, are not really compostable and have hidden plastics, making them expensive and inconvenient to recycle or compost. Nobody is really separating this plastic from the paper. However, the need for flexible and single-use materials will not go away. We need disposable packaging to wrap our food or buildings or agricultural operations or shipping consumer goods, among other applications. Our solution is in nature. Our material, the one you see in this picture, might look like plastic, but it's made with recycled natural fibers and no harmful chemicals. At Atacama, we source natural fibers and other biomasses and process them with our manufacturing robots into compostable biomaterials that degrade in high-quality soil, feeding new biomass in a perfect circular economy model. Our solution has two innovative components: the revolutionary biomaterial you already saw, and second, a packaging robot that converts our material into customized packaging at our clients' facilities whenever they need it.
To be commercially viable, materials are evaluated through performance, cost, and impact. One of the drawbacks of biomaterials is that they are weak and not durable. We have tested our material at MIT labs, and we have demonstrated that it has a strength equal or superior to industry requirements for packaging. We also proved that it has 2 years of shelf life in good conditions. Regular plastics stay in the ground for centuries or millennials. Our material is decomposed by worms in just 2 months. Finally, paper has no outdoor resistance. Our material can be left outside for weeks. Natural fibers, our feedstock, has high variability, and we use machine learning to achieve consistent quality through the performance of all of our products. Our raw materials are low-cost organic waste sourced locally today by the ton everywhere in the world, with a low-cost mechanical manufacturing process that uses low energy and doesn't include any chemical synthesis, plus on-site production, we made our material uniquely cost-effective. Finally, very important, in terms of environmental impact, our material only uses raw materials that are FDA recognized as safe to be in contact with foods, humans, and the environment. Our material is easy to sort, children can do it, and has a clean end of life. And if you don't want to compost it, which we totally understand, the city can do it for you in a cheap and easy way. Our aim is to eliminate 2.5 gigatons of CO2 by 2030. So you can see that we are the first and only scalable solution with competitive performance, cost, and impact to replace disposable plastics. Our revenue will come from selling the biomaterial cartridges to the client with a profit margin of 68%, and from leasing the equipment to them with an addressable market of $45 billion US. Our immediate application and beachhead market are packaging envelopes for clients such as the Amazons, the FedEx, and UPS of the world. These envelopes are strong and durable, cost-effective, and conveniently disposable for consumers. It took us 3 years to develop our material and robot at MIT. We went through several incubators, and we're happily here at Nucleate, launching our company. We're currently working with Wilson Sonsini to fully protect our IP, and we look to expand our pre-seed award of $150K with $150K to launch 2 pilots later in the year with Boston retailers and start selling mid-next year, first here in the US and then in developing countries. After we deploy our first product, Jose Tomas and I will continue developing robots and AI to accelerate the adoption of sustainable and bio-based materials, supported by an advisor dream team from law, policy, packaging industry, business, and operations. It is time for Atacama to blossom again. With you, we can make a difference today. Thank you.
All right, amazing. Hand it over to the judges immediately. Can we get the judge mic live, please?
There we go. Great. Good to see you. Awesome presentation. Just wondering, so the inputs, so where do you see the pieces that are differentiated? Is it in the material inputs, and how do you source those? And then also it sounds like it's really the AI robot piece, but I wonder if you could talk a little bit about like the input, and if there's something there that's also, you know, unique to you, and then a little bit more on the, what I think is the AI and robot, but really trying to understand those 2 pieces. Thank you.
Here we have 3 components. In the input, of course, the innovation, the formula of the material is novel, and the biomass it uses as feedstock besides natural fibers, of course. But the biggest innovation here, I think, has to do with the manufacturing process and the AI. First, we're tackling an issue that natural materials or renewable materials face, that is their variability. So with this AI, we can use the formulas and input, or the variations of the different biomasses in the world as input, and get a formula that will provide the same performance. But most importantly, actually, the manufacturing process. So when we developed the material, we were taking in account these 3 points that we define as the problems of current plastics. And we analyzed that scalability was the huge challenge. So we came backwards and started with a scalable manufacturing process that already existed in the industry to build upon on. And then developed a material that will fit that manufacturing process.
Thank you.
Thank you.
It wasn't clear to me what the tons of raw material is. I'll start there. You said there's tons available of your raw material. What is the raw material?
That's something we cannot say because we're working on the IP right now, so lawyers suggested not to say which components.
Okay.
What we can say is based in natural fibers.
Yeah.
Could be
Okay.
recycled natural fibers, bleached, unbleached.
In any embodiment of natural fibers plus other biomasses.
And then you were very precise on your gross margin. How confident are you on a 68% gross margin when you haven't scaled it at this time?
We base those numbers on the paper industry unit economics. So our manufacturing process is built upon that specific manufacturing process. So we work with forestry in analyzing the individual costs. Of course, that's not precise. We just like precise numbers, and that's what we got. It's going to change while we work on the manufacturing process, but it shouldn't move too much from that.
Yeah, and the last question is, I think it's a really innovative approach. But then you said that you're also going to lease the packaging equipment. Is that a requirement? Because so very different businesses, being in the automation business and the materials business. So maybe you could comment on that.
So we run into that business model this year. We pivoted from usual business model where we have a machine on site, our factory, we produce the material, and we sell directly, this mailer to e-commerce companies. What we've seen due to the supply chain problems is that e-commerce companies, and especially in packaging, because packaging is so cheap that logistics have become so expensive that most of the price goes in logistics. So companies as Amazon have started to get the equipment on site to build their packaging. So it's a new business model that has started moving on in this industry, and we are experimenting on it. In any ways, we're currently de-risking the manufacturing process, so this is a decision we can take in the scope of the next year and go which one of the two.
Hi, great presentation. A really interesting beachhead market. And I'm just wondering how much work or how much do you understand about, I think the Amazons of the world would obviously want to purchase more eco-friendly materials. They obviously have other requirements about how those materials have to perform in various weather and strength and dropping and whatever happens during shipping. And so have you done any sort of testing on how these materials form once they're in the envelope and how those compare to what the requirements are for Amazon and other sort of companies that are using these types of mailers?
So we went through I-Corps where we interviewed a lot of possible clients. That was where we defined this beachhead market. But also we built like a matrix of the specs that the material has to have in order to work as a mailer in this industry. We're constantly talking with possible clients to see what they think about the different options. But most importantly, our goal and our first pilot is all about the material. So once we finish developing the manufacturing process, we'll make a pilot just about the material to test its performance directly with clients and see what works and what not. In parallel, right now we're building a material library varying the specs to have answers to what clients or users may say. So I don't know if they need more tensile strength or puncture strength. We already have formulas that fit that requirements.
Great, thank you.
Okay, team, adding to the team, and how do you think about what you need here in this phase?
Adding to the team, we are looking to work with generally mechanical engineers, and because we have a high focus on expanding the manufacturing process of our product, and later on we plan to expand also the performance. So then it will become about R&D of the material and the chemistry of the material, etc. And we're focusing in the US as first market, and our target is to work globally. So then we see an increase in sales and market research.
Also, to our team, we'd like to add experts in supply chains. You don't need to make a lot of clients talk because there's just a few clients. If you assure 2 or 3, you got a lot of money. But you have to understand how do you fit into a complex supply chain. So hiring supply chain engineers that allow us to map and understand how we fit in will be really important.
Thank you.
Thank you.
Probably have time for one last question, if there are any questions from the judges. Okay, let's give it up for Team Atacama.
Thank you.
Thank you guys so much. Brilliant. You guys can exit.
Thank you.
Whoa.
All right. Moving on, so those were the 2 EcoTrack finalists, which is the sort of track of the Nucleate Activator program that focuses on life science solutions to non-human health applications, sustainability, clean energy, materials. Now for the last 4 finalists that will present, we're moving into the Activate Bio track. So these are going to be teams working on life science technologies to address human health. Of all the teams in the cohort, folks are working on things from therapeutics to diagnostics devices. And we're going to start with Parasite Bio. They're working on a really interesting platform to use parasite immunomodulatory biology to try and affect human health in a positive way. So without further ado, please give it up for Team Parasite Bio.
Thank you. Thanks so much. Cool. Thank you guys so much for coming. So we're Parasite Bio. We harness the immune evasion strategies of parasites to make better drugs. Oh, there it is. So our core team consists of me. I'm James DiCarlo. I'm a resident clinical pathologist and scientist at Brigham and Women's Hospital. My background is in developing some of the first CRISPR-Cas-based tools for genome editing in eukaryotes, as well as the first CRISPR-Cas-based gene drive. But over my medical training, I realized that a big problem in medicine is controlling a dysregulated immune system, which is why I co-founded Parasite Bio to really create a novel paradigm shift in how we develop immunomodulators. And I met Evan along the way.
Hi, everyone. I'm Evan Crane. I'm an MBA student at Sloan. Prior to Sloan, I was the senior manager of business development and operations at Pandion Therapeutics. I joined Pandion in the seed stage, and I worked on the company's Series A, Series B, IPO, and eventual acquisition. And in that process, I gained a real appreciation for the importance of business development in early stage biotechs. And so when I met James through the Nucleate program, and parasites working on autoimmunity, which is what Pandion was working on, it seemed like a great match, and it's been wonderful to get to know him and Ryan over the course of the past year.
And we're missing Ryan Dinza. He's currently a faculty member at Baylor, as well as a principal scientist at AstraZeneca, using large-scale genomic UK Biobank level data to look at drug targets. So he's quite busy and didn't end up traveling here for this, but he's here in spirit. And we have a really fantastic scientific advisory board. George Church, professor at Harvard Medical School, has been really great for understanding the biotech landscape. He's co-founder and scientific advisor for over 24 companies. Deborah Marks has been fantastic for a lot of our machine learning approaches, looking at sequence space and trying to identify protein functionality. She's a Harvard Medical School professor in the systems bio department. And then Sean Stoll has been really wonderful and generous with both his time. He's a transfusion medicine doc at Brigham and a great immunologist. He's been giving us some lab space to do some preliminary experiments. So we're very lucky. So Evan's going to give us some motivations.
Great, so just to, I guess, set the stage for why we're here today. As you probably know, autoimmune disease is a huge problem in the United States and around the world. And I think the bigger problem within that is that over 1 million people in the US alone have autoimmune disease that's refractory to treatment. We have several treatments that we've been using for a long time to more or less affect. Steroids are still in pretty widespread use, but they have a lot of issues with global immunosuppression. Monoclonal antibodies have been the standard of care in many autoimmune diseases for the last couple decades, but they have downsides of not great efficacy. And small molecules that have been coming into the market in recent years, such as JAK inhibitors, they work well, but they have black box warnings, and so those safety issues are a big concern for physicians. So we really believe that patients need drugs that are more precise, more powerful, and more safe, and we believe that parasites would be an ideal source for these drugs.
And the reason for that is that parasites have co-evolved with humans over millions of years to control our immune response. They do that by affecting the innate all the way through the adaptive immune response by secreting compounds, many of which are proteins, to control how the immune system sees them. They do that by secreting things like complement cascade inhibitors to control the complement cascade, a very ancient and important part of the immune system. They secrete cytokine binding proteins to soak up cytokines and affect the recruitment of innate immune cells. And they also affect the adaptive immune response by controlling antigen-presenting cells, as well as causing the expansion of regulatory T cells. Moreover, all these compounds that they secrete have evolved within humans, so they're potentially safer drugs than a lot of the drugs we have on the market right now. This is not really to read, it's just to give you a flavor of the known parasitic immunomodulators spanning cytokine binding proteins, the complement cascade, as well as affecting the adaptive immune system. There are many more than this. Parasites are the source of a few proven drugs. Bival is a really powerful anticoagulant. It comes from a leech. It's been in use for over 2 decades. It's a protein therapeutic, a derivative of a protein. Nomacapan, also a protein therapeutic derived from a tick, is a really powerful inhibitor of C5, a part of the complement cascade, and also binds leukotriene at the same time. And is in phase 3 clinical trials for autoimmune diseases of the skin and blood by a company called Akari Therapeutics, also COPD. And then parasitic worms have been used to affect inflammatory bowel disease directly in phase 1 and exploratory trials really effectively. But we believe that there are so many more untapped immunomodulators in the parasitic proteome, and that's because parasites are really diverse. They have they come from both protozoal and metazoal lineages. And they have really effective strategies for manipulating their hosts, most of which are mammals. Moreover, parasites are really understudied. Only about a quarter of parasites have their genome sequenced, and of those genomes, only about 50% of the coding sequences are well understood. So we believe that there are potentially tens of thousands of more immunomodulators to be discovered. And to do that, we have a really exciting pipeline. Sorry about the arrows, they got a little messed up. But we start from parasites, and we start from the secreted compounds, mostly the secretory glands from these parasites. We develop mRNA and then cDNA libraries, so genetic libraries, that we can then express in high-throughput screens. At the same time, the output from these screens go into a machine learning-based model for identifying features for immunomodulators. This is a really important asset for us, as well as improving our workflow going forward. The outputs from these screens can then be validated in animals and go forward either to internal clinical development or for partnering and out-licensing. So a little bit more about our library approach. So right now I've been collecting ticks, basically. I go out into the Arnold Arboretum and I collect hundreds of ticks and I grow them in the lab and extract their salivary glands in vitro. No mice were harmed, just some scraps from some dissections that I did not do. Extract their salivary glands, apply them to an in vitro screen which we've developed, which I'll talk about in the next slides, and then improve upon a model which we're developing that's based just on the sequences of these compounds to predict immunomodulation going forward. So we have an early focus in the inhibition of the complement cascade. As I mentioned before, the complement cascade is a really ancient part of the immune system. It helps recruit innate immune cells, and targeting it has been really effective for certain autoimmune diseases like PNH, atypical HUS, and myasthenia gravis. But there are many other potential applications for complement inhibitors. So right now we have a novel high-throughput and multiplexable screening methodology that we can't talk too much about because it's currently in IP discussions with Brigham. But it works quite well, and that's some of our foundational technology for screening through all these immunomodulators in the complement side. We also would like to branch out into regulatory T-cell stimulating proteins to affect the adaptive immune system. This is a really important area for treating diseases like inflammatory bowel disease, and also for helping solid organ transplant rejection. It's an area that we're currently developing. So
I'm going to touch briefly on the competition in this space. So James has mentioned Akari Therapeutics, which is one of the other companies in this space. And there are a couple others who have assets that are derived from parasites. But Parasite Bio is the only company working with parasites that is looking at the entire parasitic or mammalian parasitic proteome. And we believe that with our high-throughput discovery approach, we'll be able to identify more immunomodulators from parasites and become a leader in the space. So we've had several achievements for being a very young company. We recently won the Novo Nordisk Co-Creation Greenhouse Grant, and we're currently in the process of putting together a research agreement with Novo Nordisk. We recently won the Boston Chinese Investment Club Startup Bronze Prize. We've developed this novel complement inhibitor screen IP, as James mentioned, and he's been working on this proprietary tick cDNA library, which is basically ready to go. I'll also add that we're in the process of raising a seed round, and we would love to talk to investors about, to have you get involved. And it's, as we spin out the company more formally over the coming few months, that's one of our primary goals.
And going forward, just scientifically, we'd like to finalize this machine learning-based approach for identifying immunomodulators from sequence space, finally screen a lot of our libraries that we've been developing. And in the future, we'd like to explore kind of the combinatorial effects of these immunomodulators, sequence some of our own novel parasitic genomes, as well as kind of develop some meaningful partnerships. And with that, I'd like to thank everyone so much for coming and thank Nucleate for this really wonderful experience. It's been magical. Thank you.
Give it up for Team Parasite Bio. Awesome job, guys. All right, I will hand it off to the judges for some questions. Can we make sure the judge mic is live, please?
Yeah, we're good.
Hey.
Hi. So, really interesting concept. I'm wondering, is there a way to speed up I'll say the discovery work around parasites? Like, are there other banks? Are there other I'm not familiar with this words, but to get access to either samples for sequencing, or if you put together sequencing databases and sort of get everyone to join your band, then you already have a bigger bolus of data that you can use your machine learning algorithms on. But just how can you really, how might you ramp really that earliest phase so that you can have even more to sort of work with, especially with the idea of putting things together and sort of complexing what may be possible.
I definitely think that there are kind of a plethora of opportunities that we can harness. For one, there's a vast amount of sequences that are just publicly available. I think there are about 50 or so worms that have been sequenced so far. Of the tick genomes, there are about 12 that are publicly available. And still many of those sequences are still not understood, and those that are understood, we can start putting those into our model. Secondly, there are some repositories of organisms that they're not living. There's a tick repository, I think it's in Alabama, and it has many of the tick species that we could potentially harness. A lot of them aren't alive, so we can't do some of our fancier approaches, but we could really kind of bulk up a lot of the sequencing of their genomes. And then, so yeah, I think those are two definite approaches. So I think kind of harnessing as much as we can and negotiating with the rest of the parasite community would be something that we could do.
You mentioned that your competition, there are other competitors doing this, and that you're 1,200 versus 1, but you're not getting 1,200, I assume, from the Arnold Arboretum. And so it really is, how scalable is it? And what differentiates you from the others who are already in the field from going from 1 to much higher numbers to get to the same place?
Yeah, I think it's like a twofold approach. I think it's both our high throughput screening methodologies, the complement cascade inhibitor screening method that we have, I think is unique to us. And then just a mindset to look at these things in a multiplex high throughput way, I think is giving us a real advantage. Also, I think Yeah, so that's definitely one. I think from an asset perspective, too, we have a lot of potential for expanding the asset creation just by mining so many of these genomes. I'm trying to think what else.
I think it's basically the major ones. Right?
Yeah, I don't know if that answers
Yeah,
the question.
that's fine.
Yeah.
And if you look at any of those diseases, there's a lot of competition. So I think that's an important thing to keep in mind.
Yeah.
And also to, open your mind, if your complement screening technology is really the sort of the secret sauce, maybe looking at other modalities other than parasite derived.
I think that's a good idea too. Oh, I forgot to mention, I think our immunomodulating prediction tools, I think, is something that they're not doing. So building up both our proprietary databases for generating those models is something that other companies are not doing. So.
Yeah, but it's not a parasite specific, I presume.
So we'll use parasite sequences to build up these models. But I think another thing that you kind of point out is that there's generalizability to the model at the end of the day. We can predict potential immunomodulators that don't come from parasite proteomes. It could be broadly applicable.
Thanks for the presentation. So one of the questions, and this doesn't necessarily apply to the complement part of the project, but in terms of sort of the Treg stimulators, but one of the things that always comes up when you're taking sort of secretomes or let's call them mixes of molecules and looking for those is ultimately to define an efficient and robust drug discovery and development path, you need to know what the target is. Mm-hmm. And so as you guys think about really how to create a translational sort of pipeline from sort of the gamish to the stimulator to the mechanism. Yep. I mean, is there anything you can build into the platform that gets to that? Because that, for all of the, even the competitors you listed, it always seems to be the rate limiting step of you can see the phenomenology even at the cellular level, but you don't quite know what the mechanism is, and then that sort of ties your hands behind your back a little bit when you think about a true development plan.
Yeah, I think that's a really good point. I think one thing that we are lucky with looking at parasite-derived proteins is that there's a potential to identify multiple proteins that do a similar thing. So I think using kind of both protein similarity and using kind of higher throughput protein docking approaches and looking at either coevolution or conservation between molecules, we could get better identification of molecular pathways. So I think that's one thing that we have going for us. Yeah, that's helpful.
Thank you.
Yeah.
All right. Let's give it up for Parasite Bio. Thank you guys so much. Great job.
Thanks, everybody. So cool.
Good job, guys.
Thank you.
All right. And moving on to our next bio finalist, Tenacity. Very, very cool team coming out of the Weiss Lab at MIT, working on a new way to do genetic circuits. Let's hear it for Tenacity.
Thank you. Hi, thanks very much to Nucleate for this opportunity. I'm here representing Tenacity Therapeutics, and we work on powering up CAR-T therapies using genetic circuits. So CAR-T therapies have been a breakthrough in the treatment of liquid tumors like leukemia and lymphoma. But despite the interest in translating these therapies to attack solid tumors, unfortunately they've run into several challenges, and there are still no CAR-T therapies approved against solid tumors. And there are a number of reasons for this, and one that I'm going to focus on right now is the lack of persistence. So in the solid tumor microenvironment, T cells are constantly surrounded by target cells to attack and kill. And in combination with the immunosuppressive microenvironment of these tumors, the T cells tend to enter this state of exhaustion in which they become unable to attack and kill tumor cells. And eventually, severely exhausted T cells will apoptose or die. And so this is really limiting the CAR-T therapies against solid tumors. And our goal is to break this connection and create CAR-T therapies that can tolerate the solid tumor microenvironment without becoming exhausted and dying. And we're not alone. Pretty much every CAR-T company out there is working on this exact problem. But our approach differs in that other companies are working on solving this problem from an outside-in approach by blocking 1 or 2 signaling pathways that can cause a T cell to become exhausted. And in contrast, we work on an inside-out approach, engineering T cells to be more resilient to exhaustion from a number of different signaling pathways. So let's zoom in on a case study quickly on a target that looks really attractive in the treatment of solid tumor therapies. Mesothelin is highly expressed on a number of cancers: pancreatic cancer, ovarian cancer, and mesothelioma, and it tends to be really lowly expressed on other cells in the body, meaning that it's a really attractive target for CAR T therapies. And so UPenn thought so too, and so they pioneered an anti-mesothelin CAR T therapy for the cancers I just described. And unfortunately, this therapy failed because all these patients' engineered T cells had died after 1 month. They tried delivering it another way, again, failed, T cells undetectable after 1 month. They tried using a combination therapy to increase the persistence, failed after 1 month. They even tried using some genetic knockouts to try to increase that persistence. Once again, totally dead after 1 month. And yet, despite all these phase 1 failures, as I mentioned, mesothelin is a really attractive CAR T target. So obviously, we need to work on getting these T cells to survive longer than 1 month in the body so that we can hope to have efficacy against these cancers. Luckily, there have been some really exciting preclinical studies that are starting to show us ways that we can increase persistence and reverse exhaustion. And that is through the phenomenon of transient rest, so giving the T cells a period of time where they're resting and unable to attack the tumor. So in this first study, they rested the T cell by turning CAR expression off for a 4-day period. And what they found is that when T cells were resting, they actually reversed their exhaustion to baseline levels as measured by single-cell RNA sequencing. In another preclinical study, they modulated the CAR's ability to engage with target cells. And what they found is that after turning the T cells back on after a period of rest, there was intense proliferation. And this is in a model where all the T cells are dead after 3 weeks without this rest. So given this exciting preclinical data, we built our own version of this sort of rest phenomenon. So in the genetic circuit that we've engineered, patients take an orally available generic drug to turn their T cell population between the attacking state and the resting state. And by toggling the T cells between attack and rest, we hope to also increase the persistence of our T cells. So at this point, you're probably asking, why would I use your method over the other methods you just presented? And it all comes down to this period right here. What do you think happens when every single engineered T cell in the body is turned off from attacking the tumor? Unfortunately, the tumor is going to regrow and metastasize during that time. And that's why all the approaches that I've just described are not clinically viable. So where our advantage comes in is that we are actually able to engineer 2 populations of T cells that switch off between attacking the tumor and resting in response to these small molecule drugs. And the breakthrough technology that enabled this is that we've developed a platform to regulate gene expression by regulating RNA stability. So if we start with an RNA with a normal amount of stability, we can use a regulator protein to degrade that RNA, causing it to be unstable and turning gene expression off. And conversely, if we engineer an RNA to start out unstable, we can use that same regulator protein to stabilize that RNA, turning gene expression on. And we've dubbed this the Programmable and Nucleolytic RNA Scission-Based Stability Tuning, or PERSIST platform. And I'll go into the advantages of our platform in general in just a minute. Now, the advantages of our approach using synthetic biology, as opposed to the other advantages that I described, in which an infused drug or protein directly interacts with the CAR. And we have a number of advantages over that strategy. So first of all, our platform is able to respond to multiple FDA-approved, off-the-shelf, orally available pills. And we can very easily swap out which pills we want our therapy to respond to. Second of all, we've developed a library of regulator proteins that are each able to turn gene expression on or off at different levels. So by choosing a regulator protein, we're able to choose how high we want CAR expression to be in the on state and how low we want it to be in the off state. And then finally, as I mentioned, the same regulator protein can turn the CAR on in one population and off in the other. And that's how we're able to develop the 2-population approach that I just described. So this is our enabling technology that will allow us to increase T-cell persistence in CAR T therapies without those periods of complete rest where the tumor can regrow. So let's go a little more into this core technology that we've developed, our new toolbox for mammalian cell engineering. First of all, one advantage is that it's highly robust. So you can see that we're able to achieve up to a 300-fold change between the on state and the off state. Next, it's tunable. We've developed and characterized 9 independent regulator proteins with more in the pipeline, and they each have a unique profile of off and on fold change activity that, as I mentioned, allows us to pick exactly how highly we want our CAR to be expressed. And finally, this toolbox is stable over time. So in the middle, I'm showing sort of the gold standard of mammalian synthetic biology, the TET-on system. And it has great fold change between the on and the off stage. But unfortunately, it suffers epigenetic silencing very quickly. And so you can see in this graph, by the 3-week point, on average, the TET-on cell lines have lost 80% of their gene expression. And so obviously, this is not viable for therapies that we want to function for months or years in vivo. Fortunately, our persist cells on the right are able to actually completely resist epigenetic silencing up to 2 months, leaving it on par with current methods of expressing the CAR. So as a proof of concept, we've been working with regulating 2 fluorescent proteins that stand in for the CAR in our 2 different populations. So in this first set of cells, the blue pill is turning red fluorescent protein expression on, whereas the purple pill keeps it in an off state. And then representing the second population of CAR T cells, in this case, the blue pill maintains expression off, and the purple pill is what turns it on. And so obviously, we have a ways to go in terms of proof of concept using this circuit to control the CAR in primary human T cells. But given that we've validated that our platform works robustly in a wide number of cell lines, including human T cells, we're not expecting to run into too many challenges there. So as I mentioned, our first priority will be to control the CAR with our toggle circuit. Next, we would want to move into in vitro models of exhaustion and so show that our T cells are indeed able to resist exhaustion. Next, we'd like to go head-to-head against some of the anti-mesothelin CAR T cells that were tested in clinical trials and show that we're able to outperform the previous therapies. And at that point, we'd like to form a partnership with a company that already has developed an anti-mesothelin CAR T. And in partnership with them, conduct IND-enabling experiments that will allow us to get to phase 1 trials about 2 years from now. And as this is a platform technology, we're not limited to only the toggle circuit that I've been describing here tonight. So I've been focusing on the toggle CAR as that's what we would target first. We've also developed a number of other circuits with this technology, including a dimmer switch circuit, which uses integral feedback control to very precisely tune gene expressions. So for example, we could very tightly control CAR expression level on the surface of a CAR T cell, which has been shown to be crucial in setting the threshold between attacking a tumor cell with high expression, but sparing healthy cells with lower expression. And then, there are many other circuits that we could dream up. One that we're thinking about a lot is detecting cytokine release syndrome in a patient that's received a CAR T therapy and producing a therapeutic antibody to reverse that. But we're not even limited to applications in the CAR T space. We think that there are a number of other applications, such as those in manufacturing, that could also be fruitful. So with that, I'd like to go ahead and introduce the team. On stage right now, it's obviously just me. I am doing my PhD at MIT in biological engineering, and I've been working on mammalian synthetic biology. This project is based on my thesis work. My partner, Michelle, couldn't be here tonight. She's on a plane, unfortunately, but she is an MBA student with 7 years experience in investment banking. And we are also partnering with my PI, Ron Weiss, at MIT, who is a pioneer in the field of synthetic biology. So with that, I'd like to say thank you so much, and I'll take any questions.
Incredible job. Thank you so much. And can we get the judge mic live, please?
Hi. Great presentation, very, very clever, and I think obviously addressing some of the important issues in the space. One of my questions is really just on the complexity, right? Because if I understand this correctly, you need to create 2 different cell lines from the patient. engineered 2 different ways, and then give sort of 2 different pills, and not knowing what's in those pills, how have you thought about sort of a COGS perspective, a scalability perspective of something that has sort of 4 components going into the patient?
Yeah, thank you. I've definitely thought about that a lot, especially from a regulatory perspective. That's something that I think I'm going to need some more advice on. I have some meetings set up with consultants in the field and doctors that I'm hoping can shed a little more light on how that will be perceived.
Thanks, Noreen. I'm a big fan of synthetic biology. So, why, I mean, there's a lot of need for regulation, obviously, which is the core technology here. But why focus on CAR T? To me, CAR T is an area that, I don't know, seems like it's going to be outcompeted by other approaches in cell therapy, just given the complexity of manufacturing and delivery and somewhat the efficacy. So you just don't see as much. And obviously, the current CAR T players are all over existing technologies. So why not try to, gene therapy, CRISPR, other places where you could regulate? Have you thought about that?
Yeah, definitely. So one thing that sort of drew my attention to CAR T therapy is, first of all, the move towards allogeneic therapies in the field. So because we're using these regulator proteins, they're non-human proteins, and so in an autologous context, they have the potential for immune attack. But given the shift in the CAR T field towards allogeneic therapies, it's sort of the perfect opportunity to engineer foreign proteins into the cell and not face any immunogenicity consequences. And then I think the second part of this is that we have so much data already on CAR T trials in solid tumors in a way that we don't have for like CAR NK cells, for example. And I think looking at that clinical trial data is the best way to pinpoint the current failures and think about synthetic biology approaches to fixing them. So sort of a 2-part opportunity and also knowledge.
And then the other question is, you're using existing ligands, as you said, for on and off. Have you done any calculation to understand whether you'd be in the normal dose range of human use for those FDA-approved therapeutics?
Yeah. So I've dug into the literature and found serum concentrations for both of those drugs that I'm using. One, we are definitely within the limit. The other, we are just over it. And so that's something I'm hoping to understand in meetings with doctors, whether we could dose a little higher than current regimens. And if not, I think there's plenty of like yeast display or other protein engineering techniques that could help improve the affinity.
Could you say a little bit more about the other applications that you could go after? It sounded like any autologous approaches, you didn't think those would be kind of viable, but maybe talk, building on Peter's question, like what are the other possibilities like you consider? There may be even some sort of, adjacent to what you're talking about now, sort of in some other fields that could be interesting. But.
Yeah, certainly. So I've definitely been interested in the immunotherapy applications my entire PhD, so that's where I've focused a lot. I think CAR NK therapies are really up and coming, and as I said, we don't have the clinical data on them yet, but once we do, I would love to get into engineering them, especially because the manufacturing is so much easier. I think it'll just be so much easier from that perspective. In terms of biomanufacturing, epigenetic silencing is a major challenge limiting yields, for example, in production of biologics. Because we haven't been focusing on this application as much, I think I would need to do a little more needs finding to figure out what the exact regulation there would be, but it's definitely something I'm thinking about a lot.
Great, thank you.
All right, with that, thank you very much, Noreen. Let's hear it for Tenacity. You can exit stage back from you. Thank you. All right. Amazing. Okay, and now on to the next team. The next team that's going to be pitching, also in the BioTrack, InGel Therapeutics, and they're doing a very interesting hydrogel platform for sustained release of therapeutics in the eye. So with no further ado, InGel Therapeutics, take the stage. Let's hear it for them. All right.
Go next.
Hi everyone, I'm Deepthi Singh. I'm here with my partner Pierre Drommel and Patrick Zheng, and together we are InGel Therapeutics. We are here because there are 18 million Americans, patients in America suffering from retinal degenerative diseases. We have put a series of photos here showing you the disease progression, but in a nutshell, someone with RDD would actually feel like his world is shrinking one day at a time. And without a viable therapy, they dread the day in which the light is going to be taken away from them. And so we at InGel have one mission. We want to bring light back into our patient using our innovative cell therapy, which we developed in Dr. Michael Young's lab. He is an associate professor at Harvard Medical School. His first-in-man stem cell product is actually in clinical trial for treating patients with retinitis pigmentosa. It is in his lab that I met my super genius friend and co-founder, Pierre Drommel. He has a PhD in polymer science from MIT. And with Dr. Spector, we developed the core technology of InGel. But we wouldn't have been here if it was not for Patrick Zheng, who walked into our lab 8 months ago trying to recruit us for Nucleate. But we gelled so well that we pulled him towards our team. And that's because he brings this unique experience of taking the technology from the lab into the clinic. And we are really excited to get the stamp of approval from a pioneer in retinal surgery, Dr. Dean Elliott. We have also been very fortunate that we have got business advisors like Vinith and Pablo through Nucleate who have helped us develop this plan that can lead us to clinic. And together, we are ready to tackle retinitis degenerative diseases. This is a group of disease. We have highlighted some here. The most prominent one is retinitis pigmentosa and macular degeneration. The key takeaway is each of these diseases, they affect a unique cell type in retina. And so it's not a surprise that the company trying to develop a treatment are actually trying to replace this dysfunctional cells with their own cell product that can function better. And leading the way are Reneuron and J-Site. They have similar cells, but different approaches. A cell replacement approach, they actually have to go subretinal. It's a very invasive process, and it has its own complication, and that's why they failed in their dose escalation phase 2A. J-Site, on the other hand, is going to neuroprotection. They are trying to just halt the degeneration at whatever stage it is. Their delivery is very simple, but the challenge comes when you have to achieve a therapeutic dose using a heterogeneous cell population. It's a daunting task.
So to summarize, the fact that cell therapy, while being a major potential treatment, still possesses the challenges that Deepthi mentioned. We've outlined here the 4 main reasons why only a small % of those cells reach the target site. First, those 2 therapies were using a heterogeneous cell population. Second, there are high injection shear stress in the eye due to the injection cannula, which results in 80% cell death, debris, and inflammation. Third, the eye is an extremely dynamic environment. So injecting those cells in the vitreous, there will be a high diffusion to unwanted space away from the target site. And finally, as for any stem cell therapy, there are always safety concerns, such as immunogenicity, causing inflammation, and in the case of the eye, an increased intraocular pressure, or IOP. So that's with this in mind that at InGel, we have created a cellular factory secreting neurotrophic factors in the vitreous. It's a dual technology with, on one side, a pure rod-based cells to optimize neuroprotection, which is encapsulated in our biomimetic hydrogel to successfully deliver those cells. Our technology is a mix of expertise in chemistry, material science, and stem cell biology. First, we focused on the delivery vehicle. And for that, we take biomimetic material, namely gelatin and hyaluronic acid, and we functionalize them. I call them biomimetic because they are the same as the human vitreous. And this offers us a low immunogenicity. Then we take those material and we create novel hydrogels that are tailored for specific retinal cell types. This offers us not only a 3D viable environment for those cells, but also a high shear stress protection and a targeted delivery with those hydrogel attaching directly onto the retina. Then, finally, we focused on the therapeutics itself. We have the expertise and have successfully isolated a pure rod-based cells to offer optimal protection, secreting high amount of neurotrophic factors. So with all these innovations protected by our proprietary IP, we have created IGT-001, which is a stem cell matrix product to be injected in the vitreous of patients to halt the degeneration of their retina.
If I want to double-click on IGT-001 and maybe show you the most critical preclinical data we have is this one. We have injected IGT-001 in the vitreous of RD1 mice. In a few sentences, RD1 stands for retinal degeneration 1, which means that this strain of mice has a well-characterized degeneration of the retina, more specifically the photoreceptors, rods, and cones throughout time. We've looked at 3 groups: control, cells alone, our rods, and IGT-001, which is cell plus gel, and we've anatomically analyzed the retina 3 weeks after injection. What you see, those beautiful pictures here, are actually staining of the retina for cones. So what we are looking here, we are targeting cone survival. And if you split the retina between central and peripheral, those are the results we obtain. In the central retina, we had a 10-fold increase in cone survival comparing control to IGT-001. And for the peripheral, it was a 3-fold increase for the same groups 3 weeks after injection. What's extremely important to remember is that in humans, most of the cones are located in the macula, so in the central retina. That's why this 10-fold increase here in the central retina is unprecedented, has never been seen before, and that's also one reason why when we're talking to seasoned scientists, specialists in RD1 mice, they're extremely excited to start working with us on this potential treatment to halt the degeneration of the retina. So, to summarize all this technology, and if I go back to the 4 main reasons I mentioned at the beginning, InGel is actually able to address all of these challenges with IGT-001. First, we now have a pure neuroprotective rod cell. Second, we have a high shear stress protection, improving cell viability. Third, we have a targeted delivery with an attachment to the retina. And finally, we have a low immunogenicity, no intraocular pressure increase, which we have seen in all our animal studies.
And now let's talk about our go-to-market strategy. So if you look at the center, our core is really the hydrogel platform. And on the left-hand side, you've seen that we have already received millions of grants to prove that the platform can be used and customized for various cell types. Specifically, the IGT-001 is the green dot. That stands for human photoreceptor rod progenitor cells. Now, our goal is, with a really promising and strong data for our internal program, we're also thinking about external partnership. And that's why we want to go to people like Genentech, working potentially with their purple circle there. That's human retinal epithelial, pigment epithelial cells. And in terms of indications, our beachhead indication is retinitis pigmentosa because it's an orphan disease, but we have every intention to moving to other degenerative disease in the retina, such as AMD. So double-click on retinitis pigmentosa. On the left-hand side, you can see that in the US, there are about 100,000 patients with RP. And our first candidate, because of our neuroprotection strategy, we're focusing on the 20,000 patients that are in the middle to late stage. So they're just about to losing their central vision. And then on the right-hand side, if you think about all of these other comparable products, one thing to know is Luxturna by Sparks Therapeutic is the only approved gene therapy for RPE65 mutation RP. And that's about 1% of RP patients. They're priced at $638,000. So for us, if our product can work, we are quite confident that we can price it at $500,000 per patient. And with the 2 data points in hand, I think we're looking at a market size of more than billions of dollars. So this is our operating plan. We're in our pre-seed fundraising stage. You can see the light blue box. We want to raise a total of $1 million for 3 experiments. One is the IOP rabbit study because the rabbit eyes are larger and they represent, they're more similar to human eyes. And the second one we want to do, the 2 studies that we want to do after are 2 CRO confirmatory studies. Essentially, we want to take the data that my colleagues Pierre and Deepti have generated from the lab to the CROs, making a longer duration as well as adding in functional recovery. But our goal is to really go all the way to our Series A, raising a total of $40 million. And that is really what will help us to go into an IND-enabling study, as well as our phase 1/2A study in Q3 2025. And that's 3 years from now. And thank you very much, and hope you join us for bringing light to our patients.
All right, let's hear it for InGel. Great work, guys. And now I'll hand it off to the judges. If we get the mic live, please.
There you go. Great. Thank you for that good presentation. I was wondering if you could talk more about what you know today, based on the work that you've done, about the duration and sort of the cell structure and the incorporation into those back of the eye layers, trying to figure out why you're talking about preserving versus restoring. So maybe you could just talk a little bit about what you know today, on those dimensions of biology, and then restoring versus preserving.
Yeah.
Go.
This one?
Yeah.
So basically, we can do either one. So this is a structure of normal retina. The most important layer, photoreceptor, that processes light, it is made up of two types of cells that are rods and cones, right? In retinitis pigmentosa, the mutation is in the rods. The cones die not because of bystander effect. They die because they literally starve to death in absence of rods. And that is because the rods secrete that RDCVF factor, the neurotropic factor that opens the channel that allows the cone cells to have aerobic glycolysis, right? What we are trying to do is, if you halt the degeneration and preserve the cones, the patient will still have a considerable vision. If we want to do the replacement, we have to go really early on or really late. And a rate degenerative retina, it's really hostile. And so if you have a way to deliver the cells in that hostile condition and protect them from the degeneration, it's an excellent option. But if there is a way where you don't have to put the patient through that trauma, you can actually protect the vision by just delivering that neurotropic factor that is missing because of rod degeneration. And so that is our approach. We want to go really early on and prevent the cones from dying, so we are just preserving the vision with our therapy. And because it is encapsulated in a hydrogel, we can have a long sustained delivery.
So it's not that you're replacing or they're somehow finding their way into the structure, it's that actually out of the hydrogel comes the neurotrophic factors.
Yeah, so if one thing I can add is that I've met a lot of RP patients. If they have their cones left, their quality of life is actually okay. They can see things. The rods are responsible for peripheral vision and night vision. You're classified as legally blind when you start to lose your central vision.
Thank you.
Thanks for the presentation. How did you decide upon the first indications to go into? What criteria did you use?
Yeah, so I can start, but feel free to chime in. I think the number one thing that we're looking at is unmet need. For RP, there was actually a lot of unmet needs, especially because the only therapy that's on the market works for 1% of the patients. It's genotypically defined, it's the RPE65. The other thing that we look at is that our product, the rod and the encapsulated in the gel, is founded on a very proven biological pathway. So I think Deepti mentioned that. I think we have a slide here. That's the RDCVF pathway. It's published on a Cell by this guy, Josie Saha, in 2015, basically proving that essentially cones will die because of these neurotrophic factors are deprived from the rod death. So we just felt like both given the need and also because of the science, we're in a very unique position to take on this indication.
And you also mentioned that in other techniques using cell therapy that they're using a gamish of cells. What techniques have you developed the techniques to isolate the various cells, and is that proprietary?
Yeah, that is actually one of the major innovation. This is the isolation of those rods. We have been able to have a 95% pure rod population. We've also characterized their protein secretion, such as the RDCVF, which is extremely expressed in those rods. And we also have, the expertise that we've been able to do other cell types of the retina. And so that's also why we plan to have this internal program where we can target maybe retinal ganglion cells or cone photoreceptors or RPE.
And I guess one more question is, in this field, there's a lot of dead bodies, and clinical input. Have you developed a clinical advisory board, or what are you doing on that front?
Yeah, we have the head of retina service from Mass Eye and Ear, Dean, on our clinical advisory board. I also used to work at Horizon Therapeutics. We know it's a graveyard for startups. But when I met these guys, I think we're interested in such an amazing place to succeed. We're the only company that have used a biomimetic hydrogel for delivery. Everybody else uses phosphate buffer saline water, and that's our really secret sauce. If you look at the cell type, everybody is still working with RPE cells, and why is that? Well, RPE cells are epithelial cells. They're really, really tough. You throw them on the ground, according to DeepD, they grow. Whereas we're the only companies that can grow and culture photoreceptor. These are neuronal cells, and it works in our gel.
Thank you.
Awesome. All right, thank you guys. Let's hear it again for InGel Therapeutics. Fantastic job with the slide advancer. Thank you. All right, guys. Thank you for being so patient here and listening to all of these incredible pitches. There's so much technology. I know it can be a lot to process, but we've now arrived at the final pitch for tonight's showcase. 3DOE, this is a technology coming out of the Wyss Institute, working on a 3D bioprinting platform for vascularized tissue. And with that, I'll hand it off to the 3DOE team. Let's hear it.
I'm going to start this talk by sharing 3 upsetting statistics with you. First off, there are over 100,000 patients currently awaiting a life-saving organ transplant. That's more people than would fit in the largest US football stadium. Approximately 6,000 of these patients will die each year awaiting a transplant. And at the end of this talk, another patient will be added to that list. The field of tissue engineering has emerged as a way to address this organ shortage. But one of the major challenges that's still facing the field is trying to find a way to develop microvasculature structure within the bioengineered tissues so that we can provide blood, oxygen, and nutrients deep within the tissue as our normal body does. At 3DOE, we're taking this challenge head on. We've developed a technology that allows us to create personalized and vascularized bioengineered tissues that are suitable for human implantation. Our vision is to revolutionize the field of regenerative medicine by creating personalized, vascularized, and functional tissues to give our patients a second chance at life. The 3DOE technology is a combination of 3 smaller technologies. First, we have organ building blocks, which contain the cellular components of the functional tissue. We then have our Swift vascular bioprinting. This is a novel patented technology that allows us to create a microvasculature within our bioengineered tissues. Lastly, we have our suturable vascular cuff. This allows us to connect this microvascular circuit directly to the patient's own blood supply through their blood vessels. Over here, we have 2 cross-sectional images of bioengineered tissues. The green represents living cells, and the red represents dead cells. On the left, we have a tissue that's printed with just organ building blocks. You can see in the center, there's an area of dead cells, which is a result of the fact that nutrients and oxygen cannot diffuse through all of those cellular layers. On the right-hand side, you see a 3DOE tissue that's been printed with our Swift vascular bioprinting. The channel in the middle is one of the channels that we're able to print, and you can see that the entire tissue has excellent cell viability, as well as distribution of cells throughout the tissue. In the bottom here, you can see our suturable vascular cuffs that allow us to connect this microvascular network directly to the patient's own blood supply. This means when we implant the tissue, it is immediately perfused throughout the tissue. When we think about applying this technology to reconstructive surgery and organ transplantation, we see a number of benefits. First, we're able to provide tissues that have a very high cellular density, similar to the cellular density that's seen in native organs. Second, we're able to create the microvascular network and connect that directly to the patient's blood supply through our suturable vascular cuffs. Lastly, this is a highly personalized and customizable process that allows us to create tissue grafts specific for the patient and their surgeon. Here we have a brief schematic of our manufacturing process. We start by harvesting cells from a donor patient, isolating and expanding those cells, and incorporating them into our Swift printed tissues, and the tissue is then returned to the patient for implantation. We initially planned to have this at a single hospital where we have all of the manufacturing steps on site, but the image on the right shows that we have the ability to expand this to a national level through current tissue transportation technologies. Our grand vision is to develop solid organs for tissue transplantation. However, that's a very lofty goal, and so we're starting by developing simpler tissues first. As such, our initial application is to develop adipose grafts for soft tissue reconstruction, specifically for post-mastectomy breast reconstruction. This is going to be the topic of the remainder of our talk. I just want to warn you that the images on the next slide may be bothersome to some people, but we think they are essential to help you understand exactly what our patients are experiencing. 1 in 8 of US women will develop breast cancer in their lifetime. Every year, 200,000 of women undergo a mastectomy, either as a treatment for breast cancer or prophylactically if they're high risk. 3/4 of those patients will subsequently undergo breast reconstruction. The most important statistic on this slide is that 20% of them will suffer a complication due to the reconstruction. The images that we have down here are post-mastectomy, so the breasts have been removed, and then after an autologous reconstruction, which we'll discuss next. So there are 2 main types of breast reconstruction. First, we have the silicone or saline implants, which many of you are aware of. These are essentially a bag of saline or silicone implanted underneath the patient's tissues to recreate the breast shape. The other technique is called an autologous flap, or autologous reconstruction. Here, the surgeon takes tissue from the patient's abdominal wall with an intact vasculature, brings that to their chest wall, and reconnects the vasculature to the arteries in their chest. Both of these techniques have their own limitations. Regardless, they're quite concerning, leaving many patients to feel that there's not a safe, effective, or durable treatment option for them. Here we have a comparison of our 3DOE grafts with our competitors in the breast reconstruction space. As you can see, our 3DOE graft is the only one that provides vascular integration without the need for a donor site. Donor site morbidity is the complications that arise at the area where this tissue was transferred from. So in an autologous reconstruction, this is the abdominal wall, the buttocks, or the thigh. The severity of complications varies, but is most often abdominal wall hernias or chronic wounds, which often require surgical reintervention. Silicone implants and bioabsorbable breast implants also spare a patient the donor site, but they lack vascular integration. They do not have appropriate tissue handling properties. And as well, silicone implants need to be revised every 10 to 15 years. Now, if you imagine you're putting this in a 55-year-old woman, we're talking about 3, 4, maybe even 5 reconstructive surgeries during her lifetime. Additionally, silicone implants were recently restricted by the FDA in October of last year. They now require additional consents just so that women understand the risks that are associated with these grafts. So looking at breast reconstruction data from 2020, we've calculated an available market of $3.5 billion. Assuming we can capture 20% of that market, we're looking at an obtainable market of $700 million. There remains lots of room for growth here as we expand into more complex tissues. The pink circle that you see at the top here is the predicted market for the total global organ and tissue transplantation market. On the right, you can see a number of trends that have contributed to a 7% annual growth in this field. So the 3DOE team is combining expertise from two of the leading tissue engineering labs in the world. These are the labs of Dr. Lewis at the Wyss Institute and Dr. Chen at Boston University. Our core team consists of Dr. Luba Perry at the Wyss Institute. She's a senior scientist with expertise in tissue engineering. Dr. Daniel Reynolds, a postdoctoral research associate who has expertise in material science. Our business lead is Miriam Stilton. She's a first-year Harvard business student. And lastly is myself, Dr. Brandon Gaston. I'm an integrated vascular surgery resident at the Massachusetts General Hospital. We'd also like to thank Dr. Tatakonda and Dr. Beecroft, who have graciously acted as our business advisors throughout the Nucleate program. In summary, Team 3DOE is revolutionizing the world of regenerative medicine by creating personalized, vascularized, bioengineered tissues. Our initial applications are in soft tissue reconstruction, but we are confident that this platform technology will be able to revolutionize the way we think about organ transplants. Our timeline here shows that we're planning on spinning out of the Wyss Institute, and early next year, we'll be raising funding to fund our large animal trials with a goal for an IND in mid-2025. If you guys have any interest in this as an investor, a clinician, or as a patient, please feel free to reach out.
Okay, now we'll take questions from the judges.
Thank you. Very, very interesting. A couple questions. One, obviously you're taking patient cells, but how do you vascularize using patient material? That wasn't clear to me.
Yeah, so this is a novel printing technology which they've patented through the Wyss Institute. It's called swift printing, so sacrificial ink writing into functional tissues. Essentially, we start with our organ building blocks in a mold. We then use a erasable ink to print our microvascular network into the organ building blocks. We cure the organ building blocks basically to create a firm extracellular matrix, and then are able to erase the tissue or erase the ink that we printed into them, leaving behind our microvascular channels within the embedded organ building blocks. We're able to connect that to silicone suturable vessels so that we can connect our microcirculatory circuit directly to the patient's native vasculature.
Yeah, so that's not what's clear to me is are you, during the period of growing the tissue, is it connected to the patient or is it?
No, it's in just growing the cells out as you normally would in lab. Okay. And then they're combined to the organ building blocks for printing.
So how long do you think it takes from taking the cells to creating the matrix, et cetera?
We're talking about 3 to 6 months, which actually works really nicely. In the breast reconstruction world, after a mastectomy, when you're implanting a silicone or a saline implant, the patient gets a tissue expander, which sits there for 3 to 6 months to expand the skin and soft tissue enough to tolerate the implant. During that period is when we'll be growing their graft for them.
Thank you.
Very interesting technology, and not necessarily knowing sort of what you're using to expand and grow the cells. How are you controlling for growth during sort of building out the organ, and then of course no growth once it's in the body? So the growth metrics, and just thinking about sort of cancer and other potential disease states that can occur with sort of uncontrolled growth that you haven't killed.
Dysregulated cellular growth. Yeah. So fortunately, we're not modifying, at least initially, we're not modifying the cells in any ways. These are just the patient's own mesenchymal stem cells, which are harvested through a liposuction procedure. So if the cells inherently have a predisposition to turn into a fibroma, then they will. But again, this is adipose tissue, not breast tissue, so they're not at risk of developing a recurrent breast cancer. And, oh, go ahead.
No, just thank you. That was great.
Maybe you could come back and describe a little bit more on the market side, like of women today who are opting for some type of reconstruction, what you know today about like the silicone approach versus taking something from the body. Why are women sort of making those choices? What do you expect would sort of shift that market like more towards what I suspect is mostly silicone, maybe few on the other side, but kind of what do you know today?
So that's
and what do you still have to learn? Yeah.
That's a great question because I think we're uniquely positioned right now for the breast reconstruction market. Since at least 2017, we have data from the basically Society of Plastic Surgeons who have been recording all of their breast reconstructions. You're correct in knowing that the silicone implants dominate this market. However, there has been an increasing trend since at least 2017 for women choosing an autologous reconstruction, despite the risk of complications. That's because you don't need to come back 10 years later and have this revised. Each additional surgery raises your risk of complications. Okay, so women are starting to come around and realize that an autologous reconstruction is quite beneficial. And that's where we fit in. We can provide them with what they're looking for with an autologous reconstruction and spare them the donor site complications. Another one?
So when you're taking these, when you're taking these for the autologous site reconstructions and you're taking tissue from thighs and the stomach, do you ever have patients who are like, this is a win-win, like I'm getting my tummy tuck and I'm getting my breast it's like,
it
you know,
is.
right? And so they would choose this, you know,
Yeah, no, certainly.
as a double benefit.
It is a tummy tuck of sorts, but you're not getting a tummy tuck for the tummy tuck. You're getting a tummy tuck for your breast reconstruction, right? So if we need to take more tissue to reconstruct your breast, you may sacrifice the appearance of your tummy tuck, which also gives you, for these, which is not a tummy tuck again for the aesthetics of a tummy tuck, it's to harvest a large chunk of tissue. So you end up with a big linear scar just below your umbilicus, not the fan and steel incision that you see with a C-section, truly in the abdominal wall. So it's, yes, they can get a tummy tuck. Granted, we provide liposuction because how we harvest their cells is through liposuction. So instead of needing a tummy tuck, you come to us right before your mastectomy, give you a little liposuction, get your mastectomy, we'll grow this graft out for you, come back in 6 months, you got your liposuction and you got an autologous reconstruction.
So you two are a win-win. Okay, thank you.
On that note.
All right, thank you guys. Let's hear it for Team 3DOE.
Awesome. Oh, you need that? And a shake. Thanks, buddy. Awesome, which way do I exit? Is that way?
All right. And we have reached the conclusion of the Boston Final Pitch Showcase. So I just ask that everyone give one more huge round of applause for all the teams, not just the ones on stage, the ones in the breakout rooms, everyone that participated. Yeah, let's hear it. So in a moment, we're going to send off our judges for deliberation to award the first prizes for the bio and eco tracks. In the meantime, I would ask that everyone try and scan this QR code and fill out your preference for audience choice. And on that note, Ann, Peter, and Mira, if you wouldn't mind joining me on the stage for a second. We'd love to recognize your guys' donation of time. Feel free to jump up or you can go around on... So Peter, Ann, and Mira have been long-time supporters of our organization, and in fact, Mira actually, I believe, flew out here tonight, just for this pitch event. So I think we really need to recognize them for all of the time that they've donated. Please, let's give them a huge round of applause. A picture with Max?
Yeah, for sure.
Thank you, guys.
He wants to get in the picture, too.
You guys would just follow me this way. All right. Just exit that way, and then they'll take you to the deliberation room. Thank you guys so much. Okay, did everyone have a chance to scan the code? I'll leave it up for maybe 30 more seconds in case there are any last-minute stragglers. And with that, we are going to move on to our next section of the showcase, which is the sponsor recognition section. You know, none of this programming would be possible without, you know, the generous support of many of the sponsors that we have at the sort of event, regional, and national level. And to tell you a little bit more about our sponsors and how they're committed to supporting the next generation of bio-entrepreneurs, I'd love to introduce Mandy Williams to the stage. Mandy, let's hear it for Mandy, please. So Mandy is our EVP of Finance at the national level and just a really core member of the team, and I'll leave it to her to talk about our sponsors.
Thanks, Oliver. Thank you, and hello, everybody. First and foremost, I would like to congratulate all of the Boston participants on successfully completing the Activator program and on delivering some outstanding pitches tonight. So one more congratulations for them. And on behalf of the Nucleate leadership team, I want to thank all of the Activator teams for dedicating so much time and energy to the program throughout the last couple of months, and for allowing Nucleate to be a part of your journeys. Nucleate really would not have been able to provide you with the same curriculum or resources without the generous support of its sponsors. So while the judges deliberate, I hope that everybody will join me in celebrating the sponsors who made this Boston Activator program possible. Firstly, I would like to thank the sponsors, Firstly, I would like to thank the sponsors of this final pitch event, including the E14 Fund, Harvard Business School, Adler Innovation, Twist Biosciences, and more. This celebration really would not be possible without each one of you. And in particular, I would like to recognize the contribution of the E14 Fund. Nucleate aims to make its programs and events as accessible as possible to individuals from diverse backgrounds and geographies. So we are extremely grateful for this contribution, which allowed us to live stream this final pitch event. I would also like to thank sponsors from our regional sponsors who have supported the Nucleate Boston community throughout the duration of this Activator cycle. We are extremely grateful for our platinum sponsor, Novo Nordisk, and for our gold sponsors from the Wyss Institute, Accelerate, the Engine, and the Harvard Business School. I'd also like to thank our legal sponsors, Morrison & Foerster, Cooley, and Wilson Sonsini, for their contributions to Nucleate and for providing our teams with exceptional legal support throughout the program. Finally, I want to thank Nest Bio for sponsoring a Lab Space Award, which we will announce very soon. And as you heard earlier this evening, Boston is just one of 9 cities that ran an Activator program this cycle, and we were fortunate enough to have several groups support our Activator program at a national scale. In particular, I would like to thank Schmidt Futures for supporting our open access educational mission, and I would like to thank our national platinum sponsors, Genentech, Alnylam, and Petrie. Our Nucleate would not be able to provide free and accessible educational programs without the support of all of these sponsors. And I would just like to have one round of applause to acknowledge all of our sponsors who may be in the audience or watching at home tonight. Thank you. And I am now honored to be able to announce the winners of some of our national sponsored awards from Genentech and Alnylam. And these awards are being awarded at all of Nucleate's final pitch events nationwide. The Genentech Justice, Diversity, Equity, and Inclusion Prize is, it really recognizes a team that has developed an intentional and innovative framework for implementing a JEDI strategy into their startup. And this is a $1,000 prize that will be awarded to Sun Serum. I ask that Sun Serum please stand if they're in the audience tonight. Truly such an accomplishment, and just a testament to the work that all of our teams did this year in our diversity workshop, which was a new workshop to the curriculum this year, and we are very excited to continue implementing that throughout the organization. Finally, our other national sponsored prize is the Alnylam Scientific Excellence Award, which is a $1,000 prize that will be awarded to a team whose particularly innovative technology and proposed application taken together, present a solution to a largely unmet need. This is a $1,000 prize that will be awarded to Cash DNA. Great. Congratulations to these teams. I also ask that any team who does win a monetary prize tonight, please join me at the front of the auditorium following tonight's event to receive your award. And now I will pass it off to the Boston co-managing directors, who will return to the stage to conclude this award ceremony. Thank you.
Thank you so much, Mandy. All right, guys. So we're just at the beginning of the prize awarding process. The judges are still deliberating. So actually, I don't know if Cash DNA or Sun Serum are in fact in the audience. Yes. Could you guys come up? Maybe we can just do a quick picture with you guys while the judges continue deliberating, and then we've got a few more awards to hand out. One more round of applause for both of these teams. Thank you, guys. All right, so maybe we'll do Sun Serum first, and we can maybe get a shot with the prize in the background. You take the shot with them, I'll take yeah. Oh. Guys, come on out. Cash team. Let's go. Amazing.
Thank you.
Great work, everyone. Before we proceed, I would also ask if there are any people in the audience tonight who are a mentor to one of our current teams, either this year or in the previous year. Would you mind just standing up for a second? Yeah, let's hear it for the mentors. There's a lot of people that put in time to help our teams, experts, usually in the context of a workshop. The mentors, though, are really the ones that come in, every week, week after week, and help teams really work through the sometimes conflicting advice that they can get from the various people in the ecosystem. So, would not be the same without the mentors that we've had, and we've been very fortunate to have a relatively large and super engaged mentor pool. And if you're interested in getting involved in being a mentor or an expert in our program, please talk to one of the leadership team members after the event. And to that extent, I would also like to ask that all of the Nucleate leadership team members please stand also. These people are full-time students, and they do all of this. So let's hear it for them. If if you had a good time at tonight's event, if you enjoyed the programming that we put on and the teams, that you saw pitch, I would ask that you thank the teams, but you also thank the leadership. We're putting in a lot of hours, outside of our academic responsibilities, to make awesome events like this happen, and really just to support, the ecosystem of peers that we've built. So again, from the bottom of my heart, I really want to thank all of the Boston regional leadership, all of the national leadership. We have over 400 student leaders representing more than 70 institutions in 9 cities across 6 states in the US. So it really does take a village to make everything happening. And to all of the leadership who may be watching the live stream, I would also, just from the bottom of my heart, thank you so much. You make my life much easier every day with all the hard work that everyone puts in.
Love you, Oliver. You make it happen.
Oh. Very sweet. Amazing. All right. Do you mind quickly checking with Jeff backstage? So, as you may imagine, it's not the easiest thing in the world to pick one team from each track of all of the incredible pitches that you just saw. So I would ask that everyone just sort of bears with us for a few more moments as we wrap up the conclusion of the judge deliberation process. Also, one note, we noticed that there was a bit of a snag with the Audience Choice Award. If you were looking to vote for Atacama, there was an option called Woodpack. That was their previous name. The form should be updated, but if you still haven't submitted your choice, Atacama and Woodpack are the same option. And again, we just ask that you take a moment to fill out that form because we're tallying the results live. Maybe one more category of people that I could acknowledge. I mean, there's so many people that have put in the work, but I mentioned earlier that the mentors are meeting with teams 1 hour every week to help them integrate feedback that they get from the workshops. There are also experts that we bring in, to each workshop to help teams understand and evaluate the various aspects of venture risk around their hypotheses. So I would also ask that if there are any experts in the audience who have participated in this year's program or previous year's program, that you would also stand and we could give you a round of applause. Would really appreciate that as well. Yeah, let's do it. Amazing. Great. I think we still need a few more minutes, so maybe we could actually get all of the Nucleate leadership up on the stage for a quick second and Max, we could take one picture with the letters, and I think after that we should probably be wrapping up the process. So if you're a part of the Nucleate regional or national leadership, I would ask that you join me on the stage, take some pictures. All right, guys, we are going to take a 5-minute break as the judges wrap up their deliberation. I would ask that if you guys want to use the bathroom quickly and come back in 5 minutes, we will be announcing the first and second place winners as well as the audience choice. All right, everyone. Thank you so much for your patience. If you could take a seat, we will get started. All right, guys, we are at the conclusion of the program. Thank you for your patience. The judges put in a lot of effort and thought into their final decision. And at this moment, I would actually like to ask the co-MDs and the judges to join me on stage as we announce the award process. Come on out. Thank you very much. Big round of applause. Peter. Mira, and thank you. Yeah. All right. Slide advancer.
We're just bringing up the slides now. So give us one moment here. There we are. All right. Did this one. Okay, awesome. So the last of our sponsored awards tonight, very excited to announce the Hatch at NestBio Award. Just to remind everyone, this award is going to give this team 6 months of free lab space at the NestBio Labs in Kendall Square. And the winner is BioSense 8. Yeah, come on down. There we go. Nice suit.
All right, thanks.
Let's get a picture with Max.
Yeah.
Awesome.
Quick shout out to Nodar. Nodar, thanks for the technical help, and Steve Grant for the business side. I think he had to leave soon, but thank you so much.
Incredible. All right, I'll hand it to Jeff to present the next award.
We may have finally reached the end of our batteries tonight, so if I can ask the help of the good folks in the back to advance the slide. Excellent. So the next award that we have for you, you've actually given us all the help in selecting. So the next award is the Audience Choice Award, and this will be on the bio track. So the winner of the Audience Choice Award on the bio track tonight is going to be one more Parasite Bio.
Thank you very much.
Next up, we have the ECO Audience Choice Award, and this one is going to go to Real Seafoods.
All right. Next are the program awards, one for bio, one for eco. I would actually ask that after we announce each program award, the teams wait until we're done with both of them, and then we can get the bio and eco teams in one big picture. So without further ado, the Nucleate Bio Program Award for 2022 goes to Dorivac. All right, I'll pass it to Rebecca for the ECO Program Award.
Awesome. Thanks, Oliver. So, for the Nucleate Eco Program Award, wild microbes.
We would ask if Wild Microbes or Dorivac teams are present, just to come to the stage for a quick picture. They may not be. That's okay. We'll move on. All right. The next ones are the first place, right?
Now we come to our first place awards, and for these awards, we'll pass the microphone to our judges, who have, after careful deliberation, decided the winners of these awards. First one should be ECO.
Well, first of all, I think on behalf of the rest of the judges, we want to say it was really interesting to sit through those 6 presentations. And especially for me, to see the eco track this year. This is the first year we've had it. I've been doing this since inception, but that was really cool to have the eco track this year as part of it. So, in first place in the eco track is, oh, it's the big one, okay. Oops. It's first. Atacama. So, of course, I'm sure a few of you have noticed that that's not what you voted on. So, just a few words. You know, when we put our investor hats on, we look at, you know, clarity of market, clarity of execution plan, you know, how well is thought out the next steps and where you're going. And we felt that the Atacama team had really done a lot of that. The real seafood stuff is extremely interesting. I'm rooting for you guys because I need more omega-3s. Hopefully those fish make omega-3s. But we felt that the Atacama team really came in first place here from their presentation. So thank you. Congratulations.
Yes.
Great work. Congratulations. Congratulations.
So we move on to the BioTrack, and every year this is more challenging. First of all, because there's more to choose from, but second of all, because you're comparing, you know, product companies to platform companies, very different stages. All were extremely interesting, and all have a great potential. Here we go. First. What we felt was first place in the BioTrack was InGel. And this basically came down to the combination of having a clear product with all the elements defined and a clear path to actually testing that hypothesis. As we discussed in the questions, there's certainly headwinds there, but we felt that in terms of those criteria, that's why we gave this InGel first place. Congratulations.
Congratulations. Congratulations. Congratulations.
I just want to thank, again, Peter, Mira, and Anne for donating their time and really taking the time to think carefully about these teams and offer their feedback and perspective. And for all the teams that pitched tonight, we'll be sure to get you a copy of the judging rubrics so that you can, you know, take that advice to heart in your journey forward. So one more round of applause for our judging panel as you guys leave the stage.
Thank you so much. Thank you so much for your time. Thank you. Thank you. Thanks so much.
All right, guys, so we reached the conclusion of our program. Before we say goodnight, I would like to just bring to your attention a very exciting event that we're doing for the first time actually this summer. We're having a national summit, so this was just one of the 9 regional final pitches that are happening throughout May. But we have built this national community, as Sufyan told you about earlier this evening, and this is going to be the first time that we're going to bring everyone together. So in each region of each activator program final pitch, there is one winner, and we're going to bring everyone together in August 29th and 30th for our national summit, which will conclude with a national pitch showcase, showcasing the winners of each activator across the different cities that we're operating in. So we're very excited about that. We're also going to have some really exciting keynote speakers and guests, featured from our corporate sponsors, and hopefully the entire Nucleate leadership team will also be there. So this will be an amazing opportunity to engage with our student leaders, to engage with our teams. And if you're interested in learning more, I would just ask that you scan the QR code. There's a very short type form where you can fill out some information about how to get involved. And with that, I would just like to, again, on behalf of everyone in the organization, the judges, the teams, the leadership, the whole community, thank you so much for your time. Thank you for coming. If you're watching on the live stream, thank you for tuning in. And we hope to see you all again next year.
Thank you. Have a great night.